Good morning to all of you. So, in the yesterday we have discussed about two important cell organelle. One is the Golgi complex, and then the uh, lysosomes. So today we will continue with two more important cell organelles, and not only that, we will try to understand about some of the other things which are of I don't say that not less important, but they are in a very small quantity, including the organelles. They are not organelles, actually they are the inclusions. So first, uh, let me complete two things. One is the centrosome and then cytoskeletal elements, and afterwards we will move on to the other things. So these centrosomes, these are very important from the animal cell point of view, because they are not found in the plant cell. They are only found in the animal cell. Uh, so any animal cell, if you see, the cell has only one centrosome. Even in animal cells also there are only one centrosome. And these centrosomes generally, when the cell is in a non-dividing state, I mean when the cell is not undergoing either mitosis or meiosis, they are not visible with the what you call our light microscope, but instead of that, under electron microscope you may observe them. This one electron in the under the electron microscope, this appears like a clear spherical area. Inside this clear spherical area, you can see a pair of cylindrical structures. And this pair of cylindrical structure, which are, and both of them are at right angles to each other like this, and that is called as the centrioles. So the centriole are nothing but very short or small cylinder which contain a bundle of microtubules. These microtubules, I am going to discuss that in detail when I take up the cytoskeleton elements, but here also I should mention that these microtubules are made up of the protein what is known as tubulin. So this centrosome and centrioles are involved in intracellular or extracellular mobility. Now don't get confused with the centrosome. Centrosome is the clear spherical area enclosing a pair of cylindrical structure what is known as the centrioles. Centrosome has a pair of centriole, and both the centrosome and centriole together, they are involved in the extracellular as well as intracellular mobility. The movement within the cell, the movement, especially the nucleus and other things, and even in the extracellular, outside the cell, movement mobility is also assigned to this centrosome. So that's why they become very very important. Centrosomes is the larger structure in which you can see the centrioles. The this centrosome is also called as microtubule organizing center. What are microtubules? The component, the centrioles are made up of microtubules. These microtubules are made up of a protein what is known as tubulin. So this microtubule, it is also known as microtubule organizing center. Actually, this is an area in the cell where microtubules are produced. From here, the microtubules are produced. If you come to the animal cell, because in plant cell, the question of centrosome doesn't arise at all. But when it comes to the animal cell, centrosomes, there's a pair of small organelle. There's a pair of small organelle, the centriole. Uh, and if you see the centriole, each centriole is made up of a ring of 9 groups of microtubules. There are totally 9 groups of microtubules, and they form a ring. Uh, there's a, so there are three fused and in each group there are three fused microtubules. The two centrioles are arranged in such a way that they are, as I have told you, they are perpendicular to each other. Probably here you can see. So each microtubule, as I have told you, there are made up of ring of nine groups of microtubules. See, there are nine groups. Actually, if you count it, one, two, three, four, five, six, seven, 8 and 9, and in each group you can see three microtubule triplet you can see that. And I have told you there are two centrioles. See, both the centrioles, they are perpendicular to each other. This is how the centrioles are arranged within the centrosome. Is it clear? Yes, have you understood? Yes, tell me,
Yes, sir.
Yes, sir.
because don't get confused. This uh, microtubules, they are arranged in a ring of totally nine bundles of microtubules. I have, you can count it here, see, as the cursor moves, and each bundle consists of three microtubules. There are three fused microtubules in each grip, and as I have told you, these two centrioles are arranged perpendicular to each other within the centrosome. These centrioles usually break up into its constituent centriole at the time of cell division. Actually, these centrioles, there are two. They break up into constituent centrioles only at the time of cell division, that is during the mitosis and meiosis. Then these centrioles, they become organized into ester fibers. Suppose if you remember what you have learnt in the cell division during your plus 2 or in high schools, there you must have read the term esters, ester fiber. So these centrioles become organized into esters. Even many of the locomotory organelle, either flagellum or cilium, is also formed from the centriole or its component. So the centrioles, I have told you, they are involved in the extracellular movement, entire cell movement, by with the help of flagellum or cilia. The flagellum or cilium in the lower organisms, they are formed from the centriole or its component. Now, see, centrioles, they become organized into esters or ester fibers. Can you make out here? Microtubule on either side of the cell. Actually, both the centrioles break because they are perpendicular to each other, held together, they separate. One move to one pole and other move to the opposite pole of the cell only during the cell division. And not only they move, they get reorganized into esters, which helps in the attachment, which attaches with the chromosomes. So this is the role of the centrioles here, because if you remember in the mitosis, the chromatids are separated. The two chromatids which are held together at the chromosome, sister chromatids, get separated and move to the opposite pole during the cell division, especially during the anaphase stage of the cell division. This is achieved by the this particularly the spindle fibers or fibers. These fibers originate from this centriole and they attach to the chromosome and pull the chromosome apart, and both the sister chromatids get separated and one move to one pole and other move to the opposite pole. So this is the intracellular movement of the chromosomes is achieved by these centrioles. Okay, this is what this is the role of the centrioles. After having studied the centriole, have you understood the role? Any doubt? Anything to be asked?
No, sir.
Okay.
No, sir.
Then we can move on to the next one, that the cytoskeleton network it is. What is the cytoskeletal network? Is a network of protein filaments in the cytoplasm. It's a network of protein filaments in the cytoplasm. So these cytoskeletal elements, though we do not assign much value to it, but in the study, but they are very very important. Whatever the shape we have for the cell is because of this cytoskeletal network inside. So otherwise, holding this large amount of cytoplasm inside this thin plasma membrane would have been much more difficult than what we think. But thanks to these cytoskeletal elements, they support the large volume of cytoplasm and they render a particular shape to the cell. At the same time, the cells are flexible. Though they are flexible, can regain their normal shape because of the cytoskeletal elements. Then they also participate in large scale movement associated with the changes in cell shape and cell motility. This already I have told you while discussing the centrioles, because centrioles are also made up of cytoskeleton network of protein, that is the tubulin. So now you have studied how this tubulin protein, which is the major component of the centriole, is responsible for either the extracellular movement like the flagellar and ciliary movement, or it be the movement of the chromosome during the cell division. So they participate in large scale movement associated with the changes in the cell shape and cell mobility, and it provides machinery for organelle transport. There are sometimes the organelles are to be transported from one corner of the cell to the other, from one region to the other, and that is done by this cytoskeletal elements, including the chromosome segregation during the mitosis and meiosis. In any cell, in a eukaryotic cell, it possesses a very elaborate cytoskeleton network, and this cytoskeleton network is made up of three basic compounds. There are three different types of cytoskeletal elements, I can see that. One is the microfilaments, second one being the intermediate filaments, and third one being the microtubules. There are three different types of cytoskeletal elements. Altogether, they are responsible for the shape of the cell as well as holding the large amount of cytoplasm in that. This cytoskeletal element, see, suppose in the photograph you observe carefully, this is the entire cell. How it is able to have a square shape or a box shaped cell? This cell is box shape. How? It is because of the inner support that has been provided by different types of cytoskeletal elements, which you can see. These are the microtubules. These are the microfilaments, microtubules, microfilaments, and intermediate filaments. All together, they give a proper support inside. Otherwise, the plasma membrane would have collapsed, having such a large amount of cytoplasm within it. So, the cytoskeleton is a network of protein filaments, as you can see. That's a network of protein filaments, and they maintain the shape, and not only that, they are involved in the cellular movement. So now, see here also you can see there are different types of, these are the microtubules, these are the intermediate fibers, and these are the microfilaments. Now, what are the other functions? One is, I have told you, they are supporting shape of the cell. Apart from that, they are also responsible for anchoring the organelle. For example, if you take this the mitochondria, this is a photograph of a mitochondria. How the mitochondria can remain here only? It would have moved somewhere here. But no, it has been anchored. How? It is because the cytoskeletal elements attach to it and anchor the different cell organelle in their respective location within the cell. Now, the microtubules, they are hollow tubes and they are involved in the cell movement. This organelle occurs in pack and it assembles animals' microtubules. Whereas the intermediate filaments, they are more stable and don't break. Now let's try to understand one by one. The microfilaments. The microfilaments, they are made up of protein, what is called as actin. Actin is a very important protein, especially in the muscle protein. In the muscle there are two important proteins. One is actin and another one is myosin, which is responsible for the muscle contraction. So these microfilaments are made up of only one type of protein, which has been called as actin. The actin protein has a diameter of 5 to 7 nanometers. The diameter is 5 to 7 nanometer. And the beauty of this microfilament is they are distributed everywhere in the cytoplasm. They are distributed everywhere in the cytoplasm as an elaborate network. That means the microfilaments form a very elaborate network within the cytoplasm. What is the role of microfilaments? They support cytoplasm. They support cytoplasm and maintains texture, shape and physical characteristics of cytoplasm. So the texture of the cytoplasm, shape as well as other physical characteristic of the cytoplasm, that has been maintained by these microfilaments. Even they are responsible for the pseudopodial movement, like amoeba and leukocytes, which show amoeboid movement. That is known as amoeboid movement. So they are responsible for this crawling movement, which is shown by amoeba and leukocytes, that is the white blood corpuscles. Of course, I have told you the actin also helps in contraction of the muscles, also help in the muscle contraction. So see the how they are there. So this is the actin filament. That is the microfilaments are made up of actin. This is how they are arranged. So this actin is involved in three reasons. One is support of cytoplasm, maintains texture, shape and physical characteristics. Even they are responsible for the crawling movement of amoeba or leukocytes. It also helps in the contraction of the muscles. See here, this is another photograph which is more clear, which all of you can observe here. These are the intermediate filaments. See how they are arranged. Then the microtubules. Then the actin filament. So if you observe these different types of cytoskeletal elements under the electron microscope, they appear like this. Major component, actin filaments, microtubules, as well as the intermediate filaments, they appear like this. That's under the electron microscope. Then comes the intermediate filament. Intermediate filaments are also made up of protein, but they are made up of fibrous protein. Microfilaments, they are made up of actin protein. And intermediate filament, the second type of cytoskeletal element, it is made up of fibrous protein. The function of this fibrous protein is to provide support for orientation of various cell organelles. Within within the cytoplasm, how different organelles get oriented. To give an example, the Golgi complex which we have studied, the cis side is facing the nuclear envelope, whereas the trans side is facing the plasma membrane. How the Golgi complex is able to maintain this orientation? This is because of the intermediate filaments. So they help in anchoring as well as providing proper orientation to all the cell organelles. They are slightly bigger than the actin filament. The intermediate filaments, the fibrous proteins, they have a diameter of 7 to 11 nanometer. 7 to 11 nanometer. And these intermediate filaments, they also support the nucleus. See, if you look into the cell, the nucleus has a definite location. In some it is centrally located, in some it may be towards the periphery. How? This is because the intermediate filaments support the nucleus and helps it to anchor to their respective location. So that's why in some cells they are in the center. It all depends on the type of the cell as well as the species also. So how it is arranged? Because of these intermediate filaments which are made up of fibrous protein. See these intermediate filaments. Not only that, these intermediate filaments help the cell to withstand the mechanical stress when the cells are stretched. Mechanical strength. So even if you can press it like this, nothing happens to your cell. Again, once you leave it, it regains its original shape. Or even you can pull it. What happens? Nothing happens. Unless you pull it forcefully, very force. Otherwise, the cells can tolerate the mechanical stress. When the cells are stretched, pressed, or whatever the things. How? This is because the intermediate filaments. These intermediate filaments are flexible and at the same time they absorb this shock and maintains the physical structure of the cell. And they can span the entire cytoplasm and are anchored even up to the plasma membrane. They are found in the entire cytoplasm, they are spread and even they can anchor to the plasma membrane also. So this is the intermediate filaments, how they appear. Within the cell, the intermediate filaments are arranged like this. See, they form a network. And then coming to again the microfilaments, the actin filaments. This is a photograph how the actin appears. In the previous, you have seen how the intermediate fibrous protein appear. Here it is the actin filaments of the microfilaments. It's the most common protein. And they are concentrated just beneath the plasma membrane. These microfilaments, which are made up of actin, they are concentrated beneath the plasma membrane and give or provide the mechanical strength to the cell. The assembly of this actin filament can determine the cell shape and cause the cell movement, as I have already told you. So, suppose if they get associated with the myosin, another type of protein, they become contractile structures. Then comes the microtubules, larger component of cytoskeletal elements. larger component, because they are made up of globular protein, what is known as tubulin. You know, the centrosomes are made up of these microtubules. They are hollow pipe-like structures measuring about 25 nm in thickness, nanometers in thickness. They are hollow tubular structure, pipe-like structure measuring about 25 nanometers in thickness, and the wall thickness is 5 nanometers. It's a 25 nanometer thickness tube where the wall thickness is 5 nanometers. They are the principal components of various intercellular kinetic elements. Kinetic means moving elements. Various kinetic elements within inside the cell, like centrioles, ester fibers. They are mainly made up of microtubules. But this principal constituent of the spindle apparatus develops only during the cell division. But these microtubules, in the form of what you call the centrosome, they are not visible when the cell is in a non-dividing phase. Only when the cell is in dividing phase, they get reorganized into spindle apparatus. So these microtubules also penetrate the entire volume of the cell. Don't be under the impression that they are found only in the centrioles. No, they penetrate the entire volume of the cell. Uh, Most of the time, the microtubules radiate from the central location in the cell. The actin fibers are connected at the periphery of this microtubule. So, as we know, the main function of this is of the cytoskeletal element or microtubule is the intracellular transport as well as mitosis or cell division. So everywhere you can see the cytoskeletal elements within the cytoplasm. This is in brief about the cytoskeletal elements. Again, they are responsible for intercellular as well as extracellular movement. They are responsible for anchoring the cell organelles. They are responsible for orientation of the cell organelle within the cytoplasm. They also provide shape, texture and other physical characteristics to the cytoplasm. And they are also involved in the cell division. So these are some of the important functions of the cytoskeletal elements, which are three in number. One is the microtubules, microfilaments and intermediate fibers. Is it clear?
Yes, sir.
Yes, sir.
Now, after studying the important cell organelles, which are most of them are membrane bound, we can also study some other organelles or structures inside the cytoplasm, embedded in the cytoplasm. In that, one such thing is microbodies. These microbodies are very similar to the lysosomes because they are also bounded by a membrane, specific membrane, and they contain the enzymes. These enzymes need not be hydrolytic enzyme. What is the difference between lysosome and microbodies? Lysosomes typically contain hydrolytic enzymes, but these microbodies contain some highly specialized enzymes which are meant for a specific catalytic activity, for a very particular catalytic activity. So each microbody can contain different type of enzyme, and that is meant for specific catalytic activity. A good example is for these microbodies is peroxisomes. Peroxisome is one example for a microbody where they contain the hydrogen peroxide. What is the role of the release of the hydrogen peroxide? from these peroxisomes, they bring about oxidation reaction. So then release the hydrogen peroxide back. Like that, glyoxysomes. Glyoxysomes are found in the plant cells mainly, especially in the tissues of germinating seed. This enzyme is involved in the mobilization of energy from the stored fat seed, because in the seeds the energy is stored in the form of fat. This is the glyoxysomes which are responsible for the mobilization of energy. So these are again, these are called as the microbodies. They are small in number, and these are now. I have taken the microtubules separately after having studied these cytoskeletal elements, because this, apart from a skeletal element, the microtubules, they provide organizational structure in the interface and separate the chromosome during the cell division. This was the photograph I wanted to see. The spindle fibers, they attack and they pull the cell apart. Microtubules, hollow cylinders, of course I need not go. Tracks for. Okay, then another structure is the vacuole, which is an inclusion. These are membrane bound enclosures, vacuoles. They are also found in the cytoplasm, but more in the plant cell and very less in the animal cell. Still there are some vacuoles, so that's why I have taken while in the study of the animal cell also. So these vacuoles are classified based on their occurrence and what they contain and the role that they cater. So these vacuoles are membrane bound enclosures. That's a membrane, a sac like thing. And based on from where they occur and what they contain is more important inside. And the role that they carry out, based on that, these vacuoles are classified. So, see, in case of the vacuoles, in plant cell you can see number of vacuoles. See like this, see vacuoles. There will be numerous, but in animal cell they are less. So they look like this. These vacuoles are covered by a membrane, and that is meant for storage, maybe for even digestive enzymes or even for the removal of the waste product and even for the removal of the waste products. The vacuoles are there. Mostly they contain watery solution inside. Especially in plant, there are some plant cells where 75% of their inside is nothing but the vacuoles. So thus they help in maintaining the shape also. I have told you, based on the function, occurrence, content and the role they play, they are classified into different vacuoles. For example, the storage vacuole. Storage vacuoles are very common and prominent in plant cell. As I have told you, some of the vacuoles are so large, they occupy central large region. Sometimes 90% of the cell area is occupied by these vacuoles. In such plant cell, vacuoles are bounded by a membrane, and that is known as tonoplast. So this storage vacuoles are meant to store anything. Contain large amount of water, mineral solutes, sugars, amino acids, soluble waste substances, even gas. Even they may carry some of the colored pigments. They also act as reservoir for inorganic and organic solutes, not only water, in which along with the water they can also carry inorganic and organic solutes. Now coming to the animals, this vacuole is more important, the contractile vacuole. In almost all freshwater protozoans, you can see this contractile vacuole. The size of the contractile vacuole vary, position of the contractile vacuole also vary, shape of the contractile vacuoles also vary, but their function doesn't vary. Usually, this contractile vacuole contain water and ammonia. What is the role of this contractile vacuole? Osmoregulation. Osmoregulation. What is this osmoregulation? Maintenance of constant internal environment. That is, this freshwater protozoans, they have a problem of osmoregulation, which is totally different from the marine organisms. In freshwater, what happens? The concentration of the body fluid is more compared to the surrounding water. So as a result, more and more water enters inside the cell. Now it is the role of the contractile vacuole to pump out the extra amount of water which has entered, and thus maintain the acid-base balance inside. That is the osmoregulation. That is one type of vacuole in animals. In animals, you can also see the food vacuoles. Both in plant and animal, these food vacuoles are there. How they are formed? Suppose when the food particle enters inside through the plasma membrane, a small part of plasma membrane pinches off and packs it, surrounds it or packs it. That what we call phagocytic vesicles or food vacuoles. Yesterday while discussing the lysosomes, if you can remember, these food vacuoles merge with the lysosome inside and become secondary lysosome, and the lysosomal content then breaks the food content which is found within the food vacuole. So this is a very temporary vacuole. Once the food enters inside, a vacuole is formed around the food by wrapping the pincher of plasma membrane. Then it comes inside the cell, fuses with the lysosome, merge with the lysosome. Now the lysosome is known as secondary lysosome, and then the lysosomal hydrolytic enzymes, they digest that. These are all the living inclusions of the cell. But inside the cell, there are many non-living inclusions in the cytoplasm. Non-living, like storage bodies. Not storage vacuoles, but they are storage bodies. These are the structures which are meant to store the reserve food, more in the plant cell. They are meant to store the reserve food such as starch granules, oil globules, protein granules, which you can see in the plant cell. Like that, even in animal cell, glycogen granules, fat and lipid droplets, protein granules in animal cells, they are also stored in these storage bodies. Whenever the cell requires them, they release them back for the utilization. Till then, temporarily they are stored in these storage bodies. They are non-living inclusions. Like that, we have secretory substances or bodies also. These secretory substances or bodies, they are also vesicles. or inclusions which are filled with some secretory substances. Vesicles are inclusions. They may be secretory substances or anything secreted by the cell. More prominent in plant cell. For example, the pigments, colored pigments, even the nectar in the plant is stored in this, it comes under secretory substances or bodies. These are secreted by the cell. Even enzymes are collected. But remember, these cannot dissolve in cytoplasm. But instead of that, as soon as they are formed, they are packed in the vesicles. Either it may be secretory substances like pigments, nectar, or even the enzymes. They are stored there. Even the excretory substances or bodies. There are many things which are to be excreted out from the cell. And these excretory substances or bodies are found in the plant cell, because plants do not have excretory system. But in animal cells, animals have excretory organs or excretory systems. So whatever the waste products that are generated, they are stored as non-living inclusion within the cytoplasm, and periodically they are thrown out. Cell metabolism results in the production of lot of waste products, and those waste products are to be sent out periodically, otherwise they may become toxic. For this also, they have a special mechanism, especially in the plants. There are different types of excretory substance or bodies. For example, cystoliths. These cystoliths are nothing but the crystals of calcium carbonate. These crystals of calcium carbonate cannot be retained inside the cell. They are to be pushed out of the cell. And that is, these crystals of calcium carbonate, they are the excretory substance or bodies, and they are known as cystoliths. These are one such waste product. Then the next one is drusses and raphids. Drusses and raphids, they are also crystals of calcium, but calcium oxalate crystals. These calcium oxalate crystals, either drusses or raphids, they are also excretory substances which are the byproduct of cellular metabolism. Probably the third one is more appealing to you because all of you have seen in plants. Gums, resins, alkaloids and latex. Gums. You know, in some plants, if you cut, or even without cutting also, if there is a small injury, they ooze out some gums. A good example is the cashew tree. Some gum-like substances on the tree trunk you can observe occasionally. Or resins or alkaloids. Then even the latex, the white milky fluid, the latex, these are all excretory substances. All these inclusions, non-living inclusions, which are found within the cell, are popularly known as ergastic substances. If you want to put everything under one umbrella, you can call it as ergastic substances. That includes the excretory substances, non-living include all storage bodies, secretory substances, all these things comes under this under the one heading, that is the ergastic substances. So with this, we have completed our discussion on the cytoplasm. So if my memory goes right, we started with the structure of the cytoplasm, then we studied the different organelles starting from uh, endoplasmic reticulum, ribosomes, Golgi complex, mitochondria, and then mitochondria, then lysosomes. Then we discussed about uh, centrioles, then cytoskeletal elements, then different types of vacuoles we have studied, and then about the ergastic substances. So I hope it is clear to you. Now tell me, after having studied this entire uh, cytoplasm and their inclusion, whether you have any doubt. Yes, tell me, is there any doubt? Or if you have not followed anything, be active. Don't hesitate to. So, I am preparing a small write up about the cytoplasm and all these things. Probably within cup, yeah, by the time I complete this entire cell and its function, I will be able to give it to you. Up to plasma membrane, I have given, no? Have I given you the write up on plasma membrane? ಹೇಳಿ ಮಧು, ಲಿಖಿತ, yes, can you hear me? ಸ್ನೇಹ,
Yes, sir.
ಹಾಂ, ಅದು ಪ್ಲಾಸ್ಮಾ ಮೆಮ್ಬ್ರೇನ್ದು ರೈಟ್ ಅಪ್ ಕೊಟ್ಟಿದ್ದೇನಾ ನಿಮಗೆ? ವಾಟ್ಸಪ್ನಲ್ಲಿ ಕಳಿಸಿದ್ದೇನಾ?
ಹೌದು ಸರ್.
ಹೌದು. ಸೊ ಪಲ್ಲವಿ, ಯು ಹ್ಯಾವ್ ಸೆಂಟ್ ಇಟ್ ಟು ಆಲ್, ನೋ? ಪಲ್ಲವಿ,
Yes, sir.
then
Yes,
that
sir.
means they have not gone through it. ಸಿಂಪ್ಲಿ ಬರ್ತಾ ಇದೆ, ಏನು ಕಳಿಸ್ತಾ ಇದ್ದಾರೆ ಅವ್ರಿಗೆ, ಕೆಲಸ ಎಲ್ಲ ಕಳಿಸ್ತಾ ಇದ್ದಾರೆ. ನಾವು ನೋಡೋಣ ಎಕ್ಸಾಮ್ ಟೈಮಿಗೆ ಬೇಕಾದರೆ, ಮತ್ತೆಲ್ಲಾದರೂ ವಾಟ್ಸಪ್ ಕ್ಲಿಯರ್ ಮಾಡಬೇಕಾದ್ರೂ ಅದು ಹೋಗ್ಬೋದು. ಸೊ ನೌ ಟೆಲ್ ಮಿ ಎನಿ ಡೌಟ್ ಆರ್ ಎನಿಥಿಂಗ್ ಎಲ್ಸ್, ಬಿಕಾಸ್ ಐ ಆಮ್ ಲೆಫ್ಟ್ ವಿತ್ ಅಂಡರ್ 2 more minutes. ಐ ಡೋಂಟ್ ವಾಂಟ್ ಟು ಸ್ಟಾರ್ಟ್ ದ ನ್ಯೂಕ್ಲಿಯಸ್. Whether you have any cytology book, for this you
No,
can
sir.
refer any cytology book, it will give you all these information. And of course, I know the library is not accessible to you. So what I am thinking is, like plasma membrane, I will write a short write up for each of the cell organelle, and that will be not exhaustive. Only I will highlight some of the very important points to be noted or points to ponder, and then you have to, once the college opens, you can refer the cytology book for more information if you really need. And what else? ಹೇಳಿ. So you are following what I am teaching, because I cannot see your face, because in classroom teaching what happens, I can sense by seeing your face. But now only if you come out and tell, then only I will be able to understand. Otherwise, I have to take it for granted that the students have understood everything. So that's why feel free to interact in the last minutes, and you can open your microphone. and discuss and get your doubts clear. Okay, if not anything, probably I will stop here, and then we can